Bruce received his bachelor degree at College of William and Mary, a PhD in Princeton, and a postdoc training in MIT. Since then, he stayed in the Boston, maybe for 30 or 40 years, and now is a Stanley Cosme professor at Dana-Farber Cancer Center. As a former trainee, in Mitchell's lab, and also I'm currently faculty member at Baylor, working with Bert O'Malley. I feel it's my responsibilities to invite Bruce for this seminar series because he pioneered a lot of important discovery in nuclear receptor and cofactors, include PPAR gamma, PGC1, PRDM16. Everyone working on a deep set have to touch these three molecules. The initial study, PRD16, actually was first performed with Peter Tanas, our speaker in the last seminar series. Today, we invited Xu Xiao here to join this discussion. Also, Bruce has also made a lot of significant contribution to the field and the criminology field, identify a lot of very interesting and important hormones, include RG4, 间谍 is here, and also today's topic, So our panelists, I think 间谍, Zhou Xian, and Xinxin, they also active working on this field. I think they can discuss for hours. So I will not take a lot.
Of course, with diabetes and obesity, physical activity is very important. But what you may be less aware of is that physical activity and exercise reduce the incidence of certain cancers and is very effective for some brain disorders, including depression and dementia. And people who exercise regularly are at reduced risk for Alzheimer's disease and for Parkinson's disease. So in particular, the subject I'm going to talk about today is things that are released from muscle in the context of exercise, particularly irisin. But the idea is that with exercise, muscle may release several myokines, maybe many myokines, small molecules, macromolecules, and these ultimately may benefit the liver, the bones, the brain, the fat. And we are determined to try to identify molecules like this. So this story begins for us, this particular line of research began in 1998 when Pete Puchevert identified a transcriptional co-regulator, co-activator that we called at the time PGC1. We now call it PGC1 alpha. And we studied this in the context of mitochondrial biology, respiration, and thermogenesis. But shortly thereafter, a guy named Jiandi Lin joined the lab and made a transgenic mouse where we expressed PGC1 alpha in muscle. And of course, what we were expecting was increased mitochondrial content, and increased respiration. But that was also true, but we got a much broader phenotype like that include fiber type switching, which you can see just by virtue of looking macroscopically. The muscle is much redder. It does represent fiber type switching, mitochondrial biogenesis, angiogenesis. There are also neuromuscular junctions, fatty acid oxidation, anti-dystrophy, anti-atrophy. There have been hundreds of papers on this mouse now. And this was interesting because it was a transgenic model which looked like the mice had been chronically exercised to a very great extent. In fact, some of these changes in biology and gene program were greater even than you could do through exercise. But the thought occurred to us that this might be a very good place to identify molecules released from muscle in the context of elevated PGC1 alpha and in the context of exercise. When you exercise a mouse, you get changes in metabolites, in temperature, in the utilization of fuels. Many things are changing. And so having a sort of transgenic static system that captures some of the benefits of exercise was valuable. And we thought this would be, as biochemists, a very good place to look for new and interesting molecules that might be regulated, of course, formally in the context of PGC1 alpha elevation, but exercise as well. And about the time we were doing this work, we were not aware of it, but John Halazzi's lab at Washington University had taken an interest in what we had published. And John's lab was the first to show that PGC1 alpha is elevated in exercise. So PGC1 alpha goes up with exercise, and it induces an exercise-like phenotype in the mice. So this was a nice little exciting thing at the time. So then, using a combination of transcription profiling, some algorithms predicting prediction, and also some, I think, clever reasoning, Pontus Bostrom in the lab identified a novel myokine. In fact, I think it's still the first new molecule that had not been described before that we called irisin. And I'll tell you now what irisin is. Oh, I should also say that the biology we studied in this first paper was the browning of the adipose tissue, turning on thermogenesis. So irisin is cleaved from a type 1 membrane protein that is a single membrane spanning region with the N terminus out. And there are a bunch of basic residues in the juxtamembrane region, and Pontus cleverly predicted that this is likely to be a shed molecule. And indeed it is. So I did want to say that we did this by a combination of transcription profiling, staring at the sequences, and some good judgment by Pontus. But we wouldn't do it this way now. I just wanted to tell you that as we, work on irisin and a couple of other molecules, we also have developed some new methods, and this is actually, I think, a pretty effective method. So what we would do now is, with muscle or fat or a number of other tissues, we can slowly squeeze the extracellular fluids out from tissues using very low-speed centrifugation, 600 times G. We can get those fluids, clean them up a little bit, and then apply protein mass spectrometry. So we don't have to predict who's secreted anymore. We can just isolate the fluids and look to see what is in the fluids between the cells. Now, there's some very good things about this method and some less useful things about this method. So you all might be familiar with another method that's been developed called TurboID, which winds up putting a biotin on proteins through the classical secretory pathway. So they get 105 proteins, we get 6,400 proteins. in our extracellular fluid. Now, there's good news and bad news. The good news is we get very, very, very deep, but we do get false positives. We get some cell breakage. We have some extracellular vesicles in that mixture, whereas the turbo ID method identifies far fewer proteins, but they're almost all bona fide secreted molecules. So what I would say is Melanie's method is very good for discovery. If you really are just trying to find new things, it's quite sensitive and really quite good. You do get false positives, though. So as an analytical method, it's not that good. You can't just say because it's in our mass spec deck, it's a bona fide secreted molecule. It isn't necessarily. So we use it as a discovery tool. So back to irisin, that was not discovered that way. We have quantitated irisin in human blood using some advanced mass spec methods, something called the AQUA method, absolute quantitation. And irisin circulates in human plasma in the range of 3 to 5 nanograms per mil, which is about 0.3 nanomolar, quite a typical level for a hormone-like molecule. It's increased in exercise, and I'm actually showing you work not from our lab, but from Chelly's lab, who also used mass spec in a slightly different way to show that exercise induces irisin in the blood of humans. I'm going to talk quite a bit about the receptor today, but let me just introduce the receptor we identified using biochemical methods, cross-linking the ligand to the receptor and then doing mass spec. And the irisin receptor appears to be mostly an alpha V integrin, a member of the integrin group, mostly alpha V beta 5. And the canonical signaling for all integrins, or nearly all integrins, is the phosphorylation of FAC. We identified these in bone cells, by the way, in osteocytes. And what I mainly want to point out here is the potency of irisin. When you have a cell that responds to irisin, like an osteocyte, you can see PFAC signaling at 10 and 100 picomolar. So irisin is pretty potent when you're kind of measuring the right output. So I'm going to talk mainly today about our work in the brain. We are doing stuff still in bone. We are still doing stuff in adipose tissue, but I really wanted to focus on the brain. And the reason is that when it comes to unmet medical need, you will all be aware that Alzheimer's disease, Parkinson's disease, and other neurodegenerative diseases are a scourge in an aging world. And whereas some of these other disorders, bone disorders, diabetes, and obesity, there are a multitude of therapies. But for human neurodegeneration, it's a very poor picture where there are not many alternatives, and certainly none that are very effective. So I want to first introduce one paper by Christiane Rahns' group in collaboration with us, and just describe the methodology, because we're gonna use some of the same methodology here. What we do is we make an AAV8 that expresses cleaved irisin or GFP. We inject it into the tail vein. It gets taken up by the liver. And then over the next couple of weeks, the levels build up. And then we do whatever behavioral tests or functional tests from there. So in this particular experiment that Christiane did in collaboration with us, is tail vein inject, train the mice on a Barnes maze, wait 50 days and see whether the mouse remembers how to get out of the Barnes maze. So the Barnes maze is about 20 black spots. One of these is a hole that the mouse can escape. Mice hate being in an open field, and they would like to escape. And so after a training period, they learn the orientation of the maze in the room, and they get very good, much better at escaping. But if we, in fact, wait 50 days, the mouse that got irisin in the blood elevated, about a 10-fold elevation, remembers their training period much better than those that got GFP controls. Another point I need to make, and obviously it's implied in what I just said, is irisin crosses the blood-brain barrier, which many polypeptides do not. So this shows you the levels of irisin in the plasma. With this viral method, we get about a 10-fold increase above physiological. And if you look at the RNA, the viral RNA is detected in the liver, but not in other peripheral tissues, and certainly not in the brain. However, when you perfuse the mouse, you can measure the level of irisin in the brain and in the liver, where it's coming from, but in no other tissues that we studied. So the main subject I wanted to talk about is Parkinson's disease. So you know that Parkinson's disease, I believe, is the most common neurodegeneration in humans. It's mostly noted for motor difficulties, difficulty walking, shuffling gait, tremors in the hands and the legs. And about half of patients eventually have cognitive disability as well. In the last 10 or 15 years or so, it has been noted that Parkinson's patients almost always have aggregates of a protein called alpha-synuclein. Alpha-synuclein assembles oligomers, fibrils, and then they form what are called Lewy bodies in the nerves. And these Lewy bodies are mostly made up of alpha-synuclein. And also, very good and effective systems have been introduced in both cultured cells and in vivo, whereby alpha-synuclein fibrils, preformed fibrils, PFFs, can be introduced into cultured neurons, where they will take up the alpha-synuclein and then begin to aggregate their own alpha-synuclein, or in vivo, where alpha-synuclein preformed fibrils can be injected into the brain, not the substantia nigra, the part that is basically lost neurons in Parkinson's, but in a different part of the brain, and it'll spread prion-like across the brain. So let me show you the first experiment. Project done with Ted Dawson's lab at Johns Hopkins. And what Tien Kam did in this first experiment is to introduce PFFs into primary cortical neurons in culture. And over 7 days, the neurons will take up that alpha-synuclein and it'll seed aggregation of the cell's own alpha-synuclein. And then after a given period of time, 7 days, the cells are extracted with non-ionic detergents and then eventually with SDS. And the material that only solubilizes with SDS is referred to as the insoluble fraction. And that material is often, usually phosphorylated at position 129. So you will note here, this is what the aggregated alpha-synuclein after a week looks like. If you don't add PFFs, you don't get any. If you add the preformed fibrils, you get lots of aggregated alpha-synuclein. And amazingly, if you titrate up in nanograms per mil, irisin, even at 5 nanograms per mil, which is close to the physiological level, you can see a tremendous reduction in aggregated alpha-synuclein that you get from those cultures. And it eventually, with higher doses, it goes down almost to zero. And these are quantitated here, both the phosphoalpha-syn and the total alpha-syn. So we also did this experiment in vivo. Again, the protofibrils are injected, in this case, into the striatum. And then it spreads prion-like across the brain, eventually damaging the dopaminergic cells of the substantia nigra. So the functionality is measured in a pole test and a grip strength. the pole test, a mouse is allowed to grab a middle of a pole, then it's turned vertically, and it takes strength and coordination for the mouse to crawl down the pole. That's timed. And you can see that the mice that received the preformed fibrils of alpha-synuclein have a much reduced ability to climb down the pole, which is an increase in time, the latency time. However, remarkably, the same PFFs, if the mice were given irisin several weeks after the start of the experiment, and all this is scored 6 months after the PFFs are injected and almost 6 months after the irisin is given, you can see basically essentially complete loss of the negative function of the alpha-synuclein. Same thing with grip strength, which is more self-explanatory. That's pretty much ablated by irisin. And when you stain for tyrosine hydroxylase in the substantia nigra, if you look at these top panels, this shows you the loss of dopaminergic cells due to the action of the alpha-synuclein. And in the mice that got irisin, there's a very strong preservation of those dopaminergic cells. So all this says, both histologically and functionally, that irisin really strongly interferes with the development of this Parkinson-like syndrome. And again, I mentioned it crosses the blood-brain barrier from the periphery in the model that we are using. Much like I showed you with cells in culture, remarkably, when you look at the aggregated alpha-synuclein, this is now microdissection of the substantia nigra. You can see aggregated alpha-synuclein, and the mice that got irisin and alpha-synuclein, there's almost a complete absence of aggregated alpha-synuclein. So we're doing this now with other models that also are showing a very nice response. Alpha-synuclein can be given in multiple different ways, and so far, so good. It really looks like irisin is very effective at blocking aggregation of alpha-syn and the damage caused by that alpha-syn. So I want to go a little bit mechanistically into what's going on. And this is a very simple experiment that kind of sets the stage for, I'll show you some more sophisticated experiments. So if one isolates the endolysosomal compartment from cells while it's undergoing treatment in vitro, you can see, again, this is alpha-synuclein, phosphorylated alpha-syn, or total alpha-syn. You can see these aggregates as I've just shown you. And once again, if you apply irisin, you get reduction in the aggregates. But interestingly, if the cells, while this is going on, are given ammonium chloride, which, as I'm sure you know, neutralizes lysosomes, interfere with lysosome function, in point of fact, there's tons of alpha-synuclein in the endolysosomal compartment, even in the presence of irisin, if the lysosome function has been interfered with. And you can see this for both the phosphoalpha-syn and total aggregated alpha-syn. So this says that this suggests that irisin is doing something in the endolysosomal compartment. And I'll give you a spoiler and tell you that it appears to be an autophagic or autophagic-like process. Before I get more mechanistic, I also want to talk about tau. And here's a really important point. In these neurodegenerative diseases, there's almost never one pathological aggregate. For example, in Parkinson's, while alpha-synuclein is very, very noted and very in the disease. And so we were anxious to ask whether irisin has activity toward tau tangles as well. It isn't just purely alpha-syn or tau. So we were curious as to whether irisin had activity specifically aimed at alpha-synuclein or whether it dealt in similar fashion with other toxic protein aggregates in neurons. So what Mu Ah, a Chinese Mongolian person in my lab, did was she developed a method of aggregating tau, introducing those into primary cortical neurons and doing a very similar experiment to what we were doing with alpha-synuclein. She checks the tangles in the microscope, and then this is the result. So shown here, these are aggregates of tau isolated from the cells. In the presence of irisin, there is almost a complete loss of the aggregated tau. And this is, again, tau is also phosphorylated. You can see the same thing when you look at phospho-tau. So this would tend to argue that this process that irisin is stimulating is not specifically aimed at alpha-synuclein alone. There may be a more generalized process going on. So then we began to study autophagy and autophagic flux in a more specific way. So the way this experiment is done, it's done in primary cortical neurons that have been given alpha-synuclein. And then we introduce a construct that expresses LC3 mCherry GFP. LC3 is a protein involved in autophagic processes. And having these two chromophores is interesting because it has a red and a green color at neutral pH, but the green color is quenched in the acidic environment of the lysosome. So this process can be followed, autophagic flux can be followed with the change in the wavelengths of the signal and the loss of the green signal in particular. And that's shown here. It's very dramatic and very clear cut. Irisin is introduced, and then over several hours, nothing happens at first. And then there's a big increase in autophagic flux, which basically, simply stated, would be the acidification of the LC3 mCherry GFP construct. And this is using a method called lattice sheet microscopy. So it's a long-term imaging procedure. We are drilling down, Mu is drilling down deeper now on this process. We're interested in the type of autophagy. We have some evidence for something called chaperone-mediated autophagy, or CMA, but this is very much a work in progress of how, what is stimulating, what is the signaling pathway involved in this, in activating this process, which is a pretty profound activation. It's pretty obvious. Now I'm going to talk to you a little bit about receptor dynamics. And aside from the biophysical interest in this, you will see that this leads us into a slightly different view of the biology as well. So integrins, which you may or may not be familiar with, a little bit different than most receptors like the FGF receptor or the insulin receptor, which are always ready to receive ligands. Integrins are not like that. They actually exist in a closed form, which cannot receive signals, cannot receive ligand signals. And then it can open up, and it's only in this open state that it can bind the ligand. So the question is, how can a small protein like irisin, which has 12,000 Daltons of amino acids, how could it do this complicated opening up and then binding? And most proteins characterized that bind integrins tend to be big extracellular matrix proteins like fibronectin that have multiple domains. So I'll take you on a brief journey. So we had published in 2018, Chad Kim, that alpha V beta 5 is the most abundant irisin receptor. And this work was all done with material that was fairly pure, 80% pure. And when we did that, we can see this binding by what's called BLI, biolayer interferometry. However, we got a little bit of a rude surprise when we purified alpha V beta 5 for structural studies, really highly pure, crystal pure. We completely lost irisin binding. So that, of course, was very aggravating at the time. But being biochemists, we just know what that means. Likely is that we lost something. There's some other factor that is required for this. And in fact, that's true. So using mass spectrometry again, we found that that component that we had purified away is a heat shock protein called HSP90, specifically HSP90 alpha. Now, what's interesting is you will be familiar with most heat shock proteins are intracellular. HSP90 has been noted for being non-canonically secreted. There's HSP90 in most biological fluids. And in fact, HSP90 had been somehow implicated in integrin function, although specifically what it had to do with integrin had never been determined, but we had no choice. We actually had to kind of figure this out. So Mu is a very good biochemist. She purified alpha V, beta 5, HSP90 to homogeneity and put together that tripartite complex. And using fluorescence anisotropy to measure the binding, we could see very clearly that HSP90 was the missing ingredient. And then with this tripartite complex, you get a KD of about 30 nanomolar, quite a reasonable affinity, and also quite a bit tighter than affinity of the famous ECM extracellular matrix ligands for this protein family. So what is HSP90 actually doing? This was a collaboration with Haisha Zhao and Alan Brown's lab, and using cryo-EM, we could investigate this. And as I mentioned, you have a closed conformation, extended closed, and eventually open conformations. And then counting multiple particles, Haisha and Mu were able to show that what HSP90 is doing is moving the integrin from a closed configuration where it's unable to bind ligands, and it's opening up the receptor to allow for ligand binding. We're pretty sure, I'm not going to go through the biophysical details. Please look at the paper if you're interested. We don't believe HSP90 is touching irisin, and it isn't giving binding energy at the head group. It's actually just opening the receptor. So whereas a typical integrin ligand like fibronectin will have some domains that open the receptor and then some domains that bind to the headpiece like a ligand, in this case, irisin needs a friend. Irisin needs another factor to do that opening. And at least in the cases we've studied it, it's HSP90. Not that there couldn't be other factors in other forms of biology. Using hydrogen deuteriumic methods linked to mass spectrometry, docking models, some NMR, we eventually developed a 2.3 angstrom resolution model of irisin binding to alpha V beta 5. And the main takeaway here is that irisin does not bind at the classical binding site for integrin ligands, the so-called RGD site. So in fact, we could co-dock a piece of fibronectin that binds to the head group called FN10. That irisin doesn't bind to the same site as fibronectin. And furthermore, the receptor could bind both at the same time. And we were able to show that by the lack of competition between this piece of fibronectin and irisin. And furthermore, if we attach fluorescent probes, we could get FRET, fluorescence transfer, between this piece of fibronectin and irisin. So this is interesting because what it means is that the same receptor can function quite distinctly by binding two ligands at different faces of the receptor. And now I'm going to go back to some of the aggregation assays and show you some data that we think are quite interesting. So now we're doing alpha-synuclein aggregation, but we are applying either fibronectin or irisin, or that piece of fibronectin that binds to the head group at a different site. They both activate PFAC, which is kind of the canonical signaling pathway of integrins. So this piece of fibronectin, we've adjusted the dose for the KD binding to the receptor so we can get PFAC signaling. But whereas irisin has this activity to get rid of the aggregated alpha-synuclein, the FN10 piece has no such activity. It's binding to the same receptor, and it activates PFAC, but it can't get rid of the aggregates. Oh, I lost it. Oh, I was going to show you the same exact thing on tau, whereas irisin can get rid of tau aggregates. This piece of fibronectin also doesn't function on tau, on getting rid of tau aggregates. So one last thing, which I think is interesting and begins to tie some of the biology together with the biochemistry and biophysics. If we take a fluorescent irisin that we call irisin 647, and it's fully active, if we give it to naive cortical neurons, there's no binding or no significant binding. However, if we do the same experiment in the presence of preformed fibrils of alpha-synuclein, the irisin binding goes up and is further enhanced by giving more HSP90. So this is where the biophysics and the biology really are starting to come together. We have an HSP protein as part of the receptor complex, and the cells don't bind irisin unless they are stressed. When they're stressed with alpha-synuclein, making them, moving toward a death pathway, that's when they bind irisin. And the whole system seems as though possibly or probably through the link to the HSP90, although we haven't proven that, the system is very stress inducible, even to the level of binding irisin to the cell surfaces. So this is a model from the receptor point of view, which is that irisin cannot bind to alpha V beta 5 when it's in the completely naive state. It requires a stressor that is probably in many cases working through HSP90 alpha, but I must say that we reserve judgment on that. It could very well be that in other contexts and other cell types that maybe other factors besides HSP90 can do this job, but at least so far it looks like HSP90. And in the presence of HSP90, now the receptor is open, the head piece is available to bind, and that then gives these exercise-like effects, or specifically the beneficial effects on protein aggregates that I was showing you both in culture and in vivo. If we step back a little bit, Mu likes the Mario Brothers cartoons. So what we say is in a basal state, there's a certain level in which the Super Mario can deliver the alpha-synuclein aggregates to the lysosome. But in the presence of a stressor like the alpha-synuclein and irisin, you begin to get binding, and it's really ramping up these proteostasis pathways. So now we have 5 Mario Brothers carrying the aggregates to the lysosomes and a much enhanced ability to get rid of, at least we know now, alpha-synuclein and tau. And the work of Rudy Tanzi and Christiane Rahn have suggested that that is also true of A beta and Alzheimer's disease. So somehow the irisin signal induced in exercise is really telling some cells, at least, there's garbage coming their way, and start to get the systems together where aggregates can be removed from cells. That's a sort of a generalization, but that's sort of the picture that we have as of right now. So where are we now? So we have a collaboration with Jeff Cordover at Arizona State University. He has a non-human primate model of PD with injection of either alpha-synuclein fibrils or aggregated tau. And we are very much interested in moving this, of course, first into non-human primates and then eventually humans. So point two is kind of exciting because a lot of the current therapies you will be familiar with, for example, Alzheimer's, there's a couple of antibody treatments that are marginally effective, and they go after the A beta molecule itself. Of course, A beta is the major aggregates in Alzheimer's disease. Now, irisin seems to ramp up these proteostasis pathways and deals equally well, as far as we know, with alpha-synuclein and tau. So Jeff is an expert at developing comorbidity models. So he has both rodent and monkey models that have both alpha-synuclein and tau. And based on the data I showed you, we think it's a nice hypothesis. We're optimistic that irisin may deal with a problem that many of the other therapies have not dealt with, which is the issue of comorbidities. There isn't just one aggregate. You can't just get rid of alpha-syn. You may also have to get rid of tau, or A beta and tau. With Jeff Cordover, we're doing these experiments in rodents and monkeys using viral vectors. And Jeff is a big believer that eventually in neurodegenerative diseases, these viral vectors will be widely available. But the classic way to do this would be with a recombinant protein. Now, irisin is a very well-folded protein, but it has a very fast clearance rate out of the blood, about 30 to 40 minutes in a mouse. So we are with our little company, Avum. They are working specifically on methods of stabilization. This is a very busy area in the whole biotech world, how you take a protein, biologically interesting protein that has a short half-life, and how do you extend the half-life? Of course, in our case, it still needs to cross the blood-brain barrier. So the other approach would be other forms of irisin receptor agonists besides irisin. Remember that I told you that irisin binds to a completely distinct face from the extracellular matrix molecules. So this suggests that something like a nanobody, it may be possible to target that other face without necessarily interfering with the other classic ligands for the integrins like the extracellular matrix proteins. And lastly, and I'm not really talking about this tonight, we are working on other biological actions of irisin. In certain cancers, we see good effects. And going back all the way back to 2012, diabetes and obesity, we have a paper with Diane Mathis's lab that Mu is about to submit. We get quite good effects in diabetes and obesity, and sort of to bring the circle back to where we began. We want to show what activity irisin actually does have in diabetes and obesity. So I want to acknowledge the people involved in this, and a special shout out to these three people. Mu Ah is the biophysical biochemist in my lab that's done all the dynamics of irisin binding to its receptor and the pathways attacking the protein aggregates. Melanie Mittenbuehler did the work on developing new methods of isolating extracellular fluids and doing the protein analysis. And Christiana Rahn is still a collaborator. She was a postdoc with me. She did the first work in the brain with irisin and is doing AD work in her own lab at the Mass General Hospital. And I also want to acknowledge Chad first isolated the receptor, Shuyang Zhang and Meherhaba Yasheng are involved. Mayor Habas, a technician working with Mu. Our biophysics collaborators are Alan Brown, Haisho. John Engine's lab did the hydrogen deuterium exchange that underlied those models that I showed you. And Hari Arthanari and Christophe are very good at docking models. And lastly, our pathobiology collaborators, Christian, Ted Dawson's lab at Tyencom, and Jeff Cordover at Arizona State. And funding for this project was from NIDDK and the JPB Foundation. So thank you. And with that, I will stop my share and glad to take questions.
Yeah, let's start from the panelist. Jiande, please.
Sure.
I'm
Yeah,
sorry.
Bruce, that's fascinating. When I published the paper on PGC1 knockout mice on the topic of neurodegeneration, it wasn't my prediction that Bruce would go this far.